friends. Today we are looking at the ABO system and Rhesus system, also leukocytes. This is just a part of the blood system that we have been looking at the previous week, and it is a continuation in the cardiovascular system. So under the ABO system, we're much concerned with the blood groups, how you can group blood in different categories. And the leukocytes is just type of the cellular composition of the blood, as we are going to go further in details looking at it. So the blood group, in this case, we are saying that each and every individual contains different types of antigens on the surface of their red blood cells. And for the person to be grouped under one category or the blood group, what determines is the type of the protein that is carried on the surface of the red blood cell. In this case, the type of the antigen that the red blood cell contains. And actually, this antigen that you're talking about, it is inherited from the parents' genotypes. And as you're going to do the cross matching, you're going to find even the person is going to present with an antigen that was inherited, and it is found on the surface of the red blood cells. So each and every individual makes antibodies to these antigens that we are talking about. That's why we are saying that a person who is containing the antigen A on the surface of the red blood cells makes an antibody that is different from that this person contains. So if it's blood group A, makes the different type of antigen, which will be blood group B. And what we are saying in this case is that when a person who's with a blood group A receives the blood from a person from blood group B, it is going to react. Why? Because you find that the person with group A has got an antibody that can react with the blood group B. So that's what we are saying in this case. Each person contains the antigen with the opposite antibody. We'll get to know and find out more about what we are saying in this case. So you find that when the person receives the blood from the different blood group, there will be the signs of transfusion reactions. That can be the clumping of red blood cells. The hemolysis can take place, which is the destruction of the blood cells. Shock can result due to the reduction in the number of cells that result from the destruction of these blood cells. And we can also have the kidney failure due to the reduction in the the agglutination and the clumping of blood that is going to happen due to the fact of the antibody antigen reaction that is going to take place. So the antibodies, they circulate within the blood stream, and they've got an ability to like it is with the antigens, to associate with acquired immune system. So what we are saying in this case, the antibodies, they keep on circulating within the bloodstream because they're the one that acts as the mode of the immune system that we have in our body. Remember, the immune system is just concerned with the cells that are essential in fighting against the foreign bodies, and also we've got the antibodies that are in form of the proteins. So these are the proteins that we are talking about that keeps on circulating within the vascular system. So just like we have mentioned, each and every individual, if an individual are infused with a blood of the same group, then the person is not going to react to that blood, because you find that the body is not going to recognize that kind of the blood as a foreign invaders or the foreign body to it. So it is going to tolerate the presence of that new blood that has been transfused into him or her. But if at all given the different blood group, different blood, the person is going to react to it, because the immune system is going to mount that as a foreign body. Hence, it is going to attack and destroy those blood cells that have been introduced into this patient's body. So we're saying that the basis of the transfusion reaction depends on the donor and the recipient in terms of their compatibility. So when the blood is not compatible between that of the donor and the recipient who receives the blood, it means that the basis is going to be incompatible. Hence, one cannot tolerate the presence of the new blood that can be transfused into him or her. So there are many types of uh, collections of blood cells according to the protein that is going to be found on the surface, as we have already said, the antigen that each blood cell contains. And in this case, we are going to call it the ABO system. So the ABO system has been derived from the groups of bloods that we are going to be dealing with. There's A, there's blood group A, blood group B, blood group AB, and blood group O, making up ABO system. And we also have the Rhesus system. We're going to understand what this Rhesus system is. So for now, let's start talking about the ABO system. So we are saying that about 55% of the population are either having blood group A with an antigen A, blood group B with an antigen B, blood group AB with the antigens AB, in this case, on their red blood cells. So this is what most people have. But you find that 45% they have no any antigen on the surface of the red blood cells, and this is what we call blood group O. That's why sometimes others they call it blood group 0, because it means they've got neither blood group A nor antigen B, hence making up the group 0 of blood. So now the corresponding antibodies are called ant-A. So wherever you find this initial ant-A, just mean that it is talking about antibody A, or anti-B, antibody B. So we're saying that the blood group A individuals cannot make antigen, cannot make anti-A. Why? Because the antigen is A, and they cannot make the antibodies that are antibody A, because they can react. So now to avoid the reaction, antigen A, they make antibody B. So we're saying that they do, however, make antibody B. Blood group B individuals, for the same reason, make only antigen, or rather antibody A. So what we are saying, blood group A will make antibody B. Blood group B will make antigen and antibody B, so that there is no any reaction that can take place. Then blood group AB, they make no any antibody, because they've got both antigen A and B. Hence, if they make any antibody, it means that it can react to the cells of their own. But to avoid that agglutination or the clumping of the blood group AB does not make any antibody. Then when you look at the blood group O, they make both antibody A and antibody B. Why? Because they do not have any antigen on their surface of the red blood cells. Hence, they not tolerate any new blood that can be given to it. They are going to react and attack the antigens that are going to be found on the surface of that cell. Now, so we're saying that because blood group AB people make neither antibody A or antibody B antibodies, hence they are known as universal recipient. Why are they called universal recipient? Because they can receive blood from any person of different groups. Because we're having no antibodies, then they cannot react with any antigen that can be introduced into it. Then, so we're saying that in transfusion, either type A or B into these individuals is going to be safe, and no reaction can take place. Then the same it is with the blood group O, but in the different manner. We said blood group AB can receive blood from any person because they have no antibodies. But blood group O, they can neither be given any blood group. Why? Because they've got both antibodies. So when it receives that kind of the blood, it can react to it. But having to the fact that it doesn't have any antigen that can be reacted with another type of the blood, it is said to be a universal donor. It can be given to any blood group. It can be given to an individual with blood group A, can be given to someone with blood group B or blood group AB. Still, it is not going to react to it. Why? Because it doesn't have antigens that can be attacked by another person's antibodies. Hope you've gotten the concept here. blood group AB doesn't have antibodies to fight against antigens that can be introduced into it. But blood group O doesn't have antigens that can be reacted with another person's blood. Hence, blood group O is a universal donor. So this is just a diagram that is demonstrating what we have been talking about. So it is showing the blood group. Blood group A will have blood group antigen A that is going to be found on the surface of the red blood cells. Then antibodies is going to produce antibody B. So these antibodies, they can react when given blood group B. And in terms of the genotype, the person can either be AA or AO genotype. So what we mean in this case, the chances of this person being blood group A, it means that the parents' genotypes was either AA, both parents had A, blood group A, or they had the other parent had blood group A and the other one blood group O. When you match them together, you are having the chance of having a child with the blood group A. Same it is with blood group B. The antigen, it is antigen B. Then the antibody will be antibody A. So it can react to antigen A. And in terms of the genotype, it could be both parents were blood group B, or the other one was blood group B and the other one blood group O. Hence, high chances of having the child with blood group B. Then when you talk about blood group AB, has both antigens A and B, and by so doing does not have any antibody that can react to any type of blood, and hence the parents were blood group AB, or the genotype of this kind of the person it is AB. Lastly, we've got blood group O with no any antigen but with both antibodies, and the genotype of this kind of the person it is blood group O. So that was about the ABO system. The ABO system, in this case, we're just concerned with the blood groups. And we move on to the Rhesus system. The Rhesus system in this case is just the protein, that an additional protein that can be found on the surface of the red blood cells on top of the ABO system that we have talked about. And mostly it is known to be the antigen D, though it is just written as the Rhesus antigen or the Rhesus factor. So it is said to be, according to the research, about 85% of people they have this antigen, and for the fact that they have the antigen, they do not make the antibodies against this antigen. But there is still the probability of 15% who do not have the Rhesus antigen, and by so doing they make antibodies against the Rhesus antigen. And this does not primarily mostly make the agglutination, the clamping in any situation, but there are some occasions in which it can react. For example, in an instance of the pregnancy, for example, let's say the mother or the woman is Rhesus negative and the father is Rhesus positive. When there's when this father impregnates the woman, there's a conception that has taken place. You find that the mother is going to produce the anthraces that are going to fight against the fetus that is going to be formed with the Rhesus positive. Hence, there are chances of the antibodies that are going to be produced from the woman to destroy or to kill the baby because of the antigens that he or she is presenting with. So what we are saying, in the instance of the pregnancy, this Rhesus can react when there is no compatibility. But if both partners are Rhesus positive, there is no any antibodies that are going to be produced. Hence, the baby will be safe. And if both are negative, then it means that even the baby is not going to be positive for the mother to react to it. So that's what we are saying. Then now we're done with the Rhesus, the ABO system, and the Rhesus system. In this case, we can now start talking about the leukocytes. Remember, leukocytes is just one of the cellular composition of the blood as a tissue. In the previous time lesson, we looked at erythrocytes as being one of the cells that makes up the blood as a tissue. In this case, today we are looking at the leukocytes. So the primary function of the leukocytes, just you have to understand in this case, it is the types of the cells that have got the important function of defending the body against microorganisms and the foreign invaders that can attack the body. And in this case, leukocytes, remember, they are just white blood cells. Erythrocytes, they are red blood cells. Then platelets, they are thrombocytes. So these white blood cells that we're talking about, they defend the body against microorganisms and the foreign invaders or foreign materials that can be detrimental to the body. And we're saying that the leukocytes are largest blood cells that account for about 1% of the blood volume. So the whole blood volume of 5 liters that is found within an adult, a normal adult, the leukocytes just accounts for about 1% of that blood volume. And they contain the nuclei, and some have granules in their cytoplasm. So we are going to talk about the groups of the leukocytes, and we find that others, they've got the granules in their cytoplasm. Meanwhile, others, they do not have those granules. And the granules, they are just small structures that are found within the cytoplasm. So the first type of the leukocytes, they are granulocytes that can sometimes be said to be polymorphonuclear leukocytes because of its shapes, and there are different types of shapes. Poly means many, and the morph in this case denotes the morphology. So they've got different morphology in this group. And in this group, we've got three types of cells that falls under granulocytes. We've got neutrophils, eosinophils, and basophils. We are going to talk about them in details. And another type of leukocytes or group, it is a granulocyte. And under granulocytes, we are going to talk about monocytes and the lymphocytes. Straight away, let's talk about the granulocytes that we said that sometimes are said to be the polymorphonuclear leukocytes. What are these cells? These are types of white blood cells that are essential in the phagocytic process or in the phagocytosis, which is the destruction of the foreign invaders that can be both microorganisms or foreign bodies like just the proteins or the antigens. So during the formation of these granulocytes by the process so-called granulopoiesis, they follow a common line of development. They are going to first their stage, they are going to be the myeloblast. From myeloblast are going to develop into the myelocytes. And when they develop into the myelocytes, that's when now they are going to start differentiating into the three types that we've mentioned. And what are those three types? There's neutrophils, eosinophils, and the basophils. So in the first place, they are going just to be formed as a myeloblast, then myelocytes. From there, bone cells. That's where the production of this actually takes place under the process also called granulopoiesis. So all granulocytes have got the multi-lobed nuclei in their cytoplasm. So when you look at the structure of these granulocytes, you are going to find the multi-lobed is more like the nucleus that doesn't have the specific shape as to describe it. That's why it is just a lobed nuclei. And then you are going to see the diagram that is going to show how this is there demonstrated. And guys, we are saying that the names that you are going to give to these types of the granulocyte cells, it is going to come from their staining, how they stain when they are dipped into the dyes in the laboratory. So eosinophils, we are saying that it was given eosinophils because of this color that it takes, which is a red acid dye. The basophils takes up the alkaline methylene blue, and the neutrophils takes up both colors, red and blue, making up purple. So it appears purple. Quite right. Let's start with neutrophils. What's the function of the neutrophils as being the type of the granulocyte? It is to protect the body from foreign materials that can gain entry into it. And these foreign materials, it could be microbes with it that are bacteria, and also it could be waste materials, for instance, like cell debris. For example, if a person sustains an injury, there will be the pus formation that is going to be produced at the site of injury as time goes by due to the necrosis and other foreign microorganisms that can invade that area and forming the pus. So that pus, in this case, that's what we are calling the cell debris. And these neutrophils are attracted to the site of injury or infection by the chemical that is going to be produced by the dead cells, and this chemical is called chemotaxins. So you'll be asked, what is the chemical that actually attracts the neutrophils to the site of injury? That's what we call chemotaxins. And neutrophils, they pass through the capillary walls, since we are saying that it is actually the composition of the blood tissue. So as the blood is circulating and passes throughout the body, at the site of injury, the neutrophils are going to seep through from the blood vessels into the tissue by the amoebic movement from the capillaries or the blood. And when they reach at the site of injury, they are going to produce the phagocytic mechanism, or they are going to produce phagocytosis, which is the engulfing and killing all the microorganisms that are going to be detected at the site of injury. And in their granules, they've got the granules inside is the cytoplasm, so-called the lysosomes, which have got the enzymes that digest the engulfed materials. So for instance, let's say the bacteria has been engulfed, it's more like to coat it, to surround it actually, and it is going to produce this enzyme to digest it, now to break it down so that it can die. And the pus that may form in the affected area consists dead cells, the the live microbes, and phagocytes killed by microbes. So actually what we are talking about, through the process of phagocytosis that will be taking place, there will be actually the pus formation. So now this pus formation that is going to result from the destruction of the cells that have been invaded, and those of the microorganisms, they are going to be the pus containing all these the structures or materials. So the substance that makes up the pus actually are the dead tissues of the body, and also the dead microorganisms that have been killed by the cells, and also the phagocytes themselves. Due to the antibody-antigen reactions, others they get to be killed, and together they are going to form these pus that we are talking about here. So we're saying that there is a physiological increase in the neutrophils in some instances, and this you can be asked, what are some of the occasions, or what are some of the situations that actually increases the production of the neutrophils? Because as you'll be doing your investigations in many conditions, you'll see that when you find an elevated amount of the white blood cells, it is going to show the presence of infection. So now you need to be able to know what are those situations actually increases the neutrophils count. First thing, in the strenuous exercises, like in pregnancy, they can be elevated amount of neutrophils due to the fact of main infections that likely to occur as a result of the immunosuppressed of the person. So neutrophils are going to increase. And the number of neutrophils actually they can also increase in an instance of the microbial infection. So whenever there's microbial infection in the body, neutrophils are going to increase in number because they are the ones that are essential to fight against it. And also when there's tissue damage, so as to aggregate the inflammatory substances that are essential to combat the presence of that injury. And also in metabolic disorders like diabetic ketoacidosis, also in this case the neutrophils are going to be here in order to enhance the inflammatory reaction. In leukemia, it is going to be elevated. Leukemia, actually this is a condition whereby there are much number of immature white blood cells. So when these immature white blood cells are released within the body, within the vascular system, the neutrophils are going to detect those as foreign invaders because they are not supposed to be found in the vesicular system. Hence, they are going to increase in their number production. Then the heavy smoking, because of the toxins that are going to be found within the smoke that is ingested or inhaled, it is going to actually stimulate the production of the neutrophils. And the use of oral contraceptions or contraceptives mechanism like drugs, those pills. So when they're ingested as being the foreign bodies in the body, then the neutrophils are going to increase in their production. So these are just occasions that can increases the number or the production of neutrophils. And these actually are the diagrams that are demonstrating how those granulocyte cells appears. And we talked about having the lobed nucleus. So these are the lobed nucleus without a definite description. So if the neutrophils are first, eosinophils, and the basophils are last. Having looked at the function of the neutrophils, what is the function of the basophils? Basophils, actually in this case, they are known to be very much essential and important in allergic reactions, for they contain in their granules, as they contain something like heparin, they contain, which is anticoagulant. It prevents the coagulation of blood or the blood clotting. It also has histamine, which actually initiates the inflammatory reaction, and other substances that promotes inflammation. So these are known to be found within the basophils and hence known to be essential in allergic reaction. So we are saying that mostly the stimulus that causes the basophils to be released, it is the content. Well, for it to be released, it is actually the presence of the substance so-called allergen. Remember, with the neutrophils, we said what stimulates their arrival to the site of injury is the chemotaxin. But with the basophils, it is what we call an allergen. An allergen. That's why it is essential for the allergic reactions. So these basophils, they binds to the antibody type receptors on the basophils membrane. So this allergy, when it is introduced in the body, it is going to be detected by these basophils by attaching to it, and the basophils are going to enhance now their mechanism of attacking the foreign invaders that can be introduced into it. So a cell type very similar to basophils, there's a cell type that is similar to the basophils, but with it, it is not found within the circulatory system, and it is said to be the mast cells. So they'll ask you, what is the type of the cells that is similar to the basophils that is not found within the circulatory system, but rather in the tissues? Is actually the mast cells. So these mast cells, they've got a very exact function like that of the basophils, just that the basophils are found in the vesicular system and the mast cells within the tissues. So we're saying that the mast cells releases their granule contents within seconds of binding to allergen that can be introduced into the tissue, and which accounts for the rapid onset of allergic symptoms following exposure. For example, if at all there's a pollen that has been introduced, this allergen, when it is in the tissue, these mast cells are going to react to it. But if it's in the vesicular system, the basophils are going to take up that function. Lastly, under the granulocytes, we've got the eosinophils. Function of eosinophils, actually, it is also capable in the phagocytosis, which means the engulfing and the killing or destruction of the microorganisms that can be introduced or invade the body. But it is not that much effective like it is with the neutrophils in phagocytosis. But it is specialized in the elimination of the parasites that are large enough to be phagocytosized, because the neutrophils are just the body cells, but there are those parasites that are big in a sense that they cannot be phagocytosized. Hence, they are going to be attacked by the eosinophils, because these eosinophils, they are equipped with a certain toxin, the toxic chemical, that when it is excreted or secreted, it is going to make these actually parasites to flee away from the tissues, or rather to get eliminated from the body. Under the type of the granulocytes, we talked about, we said there are two types of leukocytes. There's a granulocytes and granulocytes. In this case, now we're talking about granulocytes. So under granulocytes, just like you have mentioned, we've got two types, monocytes and lymphocytes. Actually, they are known to have the large nucleus and without granules like those that we are talking about in the previous type of leukocytes. And they make up about 25 to 50% of all leukocytes. Just on leukocytes, it makes up about 25 to 50%, and the other percentage is for those other cells that we have talked about. So firstly, there's monocytes and granulocytes. The function of these monocytes, actually, they plays the role in the phagocytic mechanisms, just like it is with the neutrophils, just that these now they plays the role when the neutrophils they fails to yield their mechanism of eradicating the presence of microorganisms in this case. So we're saying that the larger mononuclear cells that originate in the red bone marrow. So actually the production of these monocytes is in the red bone marrow, and actually their function it is in the phagocytic mechanism. And those monocytes that leave the vascular system and found themselves in the tissues, they change or develop into what we call macrophages. They'll ask you, what form do the monocytes develop into as they leave the vascular system into the tissues? It's actually macrophages. And this is the type of the monocyte that we are talking about. That's how it appears. And this is the lymphocyte. So we're saying that both types of cells, they produce the interleukin 1. Actually, this interleukin is actually the substance that is produced within these cells, and their function, it acts on the hypothalamus so as to increase the body temperature in an instance where there's a microbial infection. That's why you see that when a person is having mycobacteria or rather bacterial infection, there will be an increase in the body temperature. Why? Because of antigen-antibody reaction that will be taking place in the body, and also the stimulation that has happened at the hypothalamus, which actually works as a thermoregulatory center. And this temperature that is elevated, it is so as to try by all means to compensate and to eradicate the presence of these microorganisms that could be the bacteria in this case. And also it stimulates the production of some globulins, which acts as the antibodies, and it also enhances the production of the activated T lymphocytes that are essential in the chronic inflammatory processes. The very interleukins also, they've got the functions of rather macrophages. They've got important functions in inflammation and immunity. Why? Because it works in a phagocytic mechanism, and also in inflammation. Remember, when you're talking about inflammation, in this case, we're just talking about the response that the body is going to produce in an instance of infection or injury. So whenever there's an infection or there's an injury, inflammation is going to result. And these are the inflammatory cells, like the monocytes and the macrophages that we're talking about in this case. So the macrophages functions in close association with the monocytes, those that are found within the vascular system, and also the lymphocytes, which actually influences its activity. And these they are actively phagocytic, and if they encounter the large amount of foreign or waste material, they tend to multiply at the site of infection or injury and wall of the area. So this is in an instance whereby if at all there's a large amount of inflammatory or infectious agents that have been introduced in the body, hence it is going to stimulate these monocytes to multiply and increase in number and to wall off the area that has been invaded, so as to result in destruction of the tissue that has been invaded with these microorganisms. And we have given an example in the lungs. If at all the person has inhaled much of microorganisms, this mechanism can happen actually, and their number are also going to increase, just like it was with the neutrophils, when there's a microbial infection, when there's collagen disease, or when there's a non-infective bowel condition, any condition that is going to result due to the fact that we said that even without an infection, but when there's just an injury, there's inflammation that is going to result. And then we say that these monocytes actually the inflammatory cells. Hence, even in non-infective conditions, the monocytes are going to elevate in their count so as to enhance the inflammatory process. And then another type of the cell that found under a granulocyte is the lymphocyte. So we're saying that the lymphocytes are much smaller than it is with the monocytes, but within the large nuclei. The lymphocytes, they circulate within the blood and much mostly in the lymphatic tissues. That's where they are going to be found. That's why its name, lymphocytes. Lympho is the lymphatic, from lymphatic, and the cytes is just the cells of the lymphatic system. So most are going to be found in the lymphatic tissues like the lymph nodes and the spleen. And we're saying that these lymphocytes actually they develop from the pluripotent stem cells in the red bone marrows, and they travel into the blood, from the blood into the lymphoid tissues or lymphatic tissues, which are like those the spleen that we have already talked about for further development. And when they develop in those lymphatic tissues, they are going to become immunocompetent, being prepared to fight against any foreign invaders that can be invade the body. So what we are saying, these cells, their production site, it is in the bone marrow. As they are produced from the bone marrows, that's when now they can go and further develop in the lymphatic tissues for them to be fully equipped to fight against the microorganisms that they can be faced with. So as they are developing to become immunocompetent, they are going to divide into two distinct types of lymphocytes, which is the T lymphocytes and B lymphocytes. T lymphocytes, these are types of the lymphocytes that are essential and that works in the cellular immunity. Then the B lymphocytes, they are types of the lymphocytes that are essential in the humoral immunity. When you say humoral immunity, this is the immunity that works with the antibodies to fight against infection and foreign invaders. And when you say cellular immunity, in this case, we mean the type of the immunity that uses the cells to fight against infection and foreign invaders, like those that we talked about in the granulocytes. Friends, thank you very much. This has been for today. We're looking at the ABO system, the Rhesus system, and also the leukocytes. I guess it's been interesting. You've enjoyed today's lesson. Thank you for your attention and stay safe. Stay away from the virus. Keep safe. It's been Fred. Thank you very much.